And the speaker is Anurag Shakharkar from University of Saskatchewan, and he will be talking about variations in gene-pair correlation as potential diagnostics in astronauts.
Okay. All right. Thank you, everyone, for being here. As said before, my name is Anurag, and I'm going to be talking about gene pair correlation and astronaut health, what we believe is the key to the metagenomic space race. So just a quick introduction. Like I said before, my name is Anu. I'm a third-year undergraduate student at the University of Saskatchewan, and this is my first research conference, and I'm very excited to be presenting here today, especially at COSPAR. Now, if I ask this question, what is the most important issue affecting astronauts today, to this room, I'm bound to get many different answers, probably depending on what all of your specialities are. But of course, there's no one right answer. There's tons of issues that astronauts face in space travel. Prolonged microgravity and cosmic radiation exposure, causing bone and muscle loss. You have immune deficiencies, impaired renal functions, neurological irresponsiveness, cardiovascular issues, you have cell proliferation, apoptosis. All of these are quite important functions that are affected, and there's no one solution to any of these issues, and also no comprehensive way of tracking health. Although there are some workarounds, here you can see American astronaut Sonny Williams running on the space station to address microgravity-caused bone and muscle loss. However, astronauts need to exercise for 2 to 3 hours a day, which is extremely disruptive and not a long-term solution. So it's very clear to see that a more comprehensive solution is needed. Instead of monitoring and addressing individual issues, why not address how the body reacts to spaceflight? And so this is where my project comes in. As with any project, we started with data collection. Before I give specifics, I'd like to take a quick second and talk about open sourcing research. Space is definitely humanity's future, and we should be working towards it together. Open sourcing research is one of the easiest and most practical ways of scientific collaboration, and it allows further insights to be gleaned from existing data and saves time and resources as well. So I'd like to thank everyone here who is open sourcing their data and encourage everyone who isn't to very strongly consider doing that. So the study this data was obtained from is titled Effects of a Closed Space Environment on Gene Expression in Hair Follicles of Astronauts on the ISS. It's definitely a mouthful, but I would encourage everyone to take a look because it's very interesting. But back to my study, the data that I'm using is microarray data, and it's accessed from the NASA Gene Lab repository, which again, cannot recommend highly enough. Moving on. Eventually, hopefully. Okay, so this image is from the previous study, and it shows their sample collection schedule. So hair samples were collected from 10 astronauts, twice each, pre-flight, in-flight, and post-flight after returning from the ISS. The collection times ranged from 6 months before leaving to 3 months after arrival back on Earth. These samples were then sequenced using microarray and real-time qPCR. After data accession, again through GeneLab, this data was cleaned, prepped, and pre-processed for a differential expression analysis, or otherwise known as DEG. Now, I'm not going to talk too much about DEG. I'm sure most of you understand the basics, and that's pretty much all that's needed. So this looks like a very boring slide, but actually to me it's one of the most interesting slides in my presentation because the amount of data that it conveys is just incredible. So I'm just going to go through a few of our most upregulated and downregulated genes here. LIMCH1, our most upregulated gene, inhibits cell migration, and it could be a possible explanation for recent results that space travel can change cancer cells into less malignant phenotypes. IER3, our second most upregulated gene, is important in cell death, inflammation, of course, and tumorigenesis, and it could be important in cancer as well. Now, going to the downregulated genes, NFATC1 was talked about yesterday in a different presentation and is involved in osteoblast and osteoclast differentiation, T-cell differentiation, cardiac valvegenesis, and we think it's one of the main genes responsible for bone loss in space. And looking at KIDINS 220, it's responsible for development and function of the nervous and cardiovascular systems. So again, all of these genes play important roles. And if you look at all of the differentially expressed genes, most of them play significant roles in functions that are affected by space travel. So of course, this DEG analysis was a great validation of existing data and a good starting point to build on for the next part, which is kind of the real meat and potatoes of this research study, which is correlation analysis. So correlation can get complicated, but I'm going to give a high-level overview just to make sure we're all on the same level. So if you want to find out the correlation between two genes, for example, gene X and gene Y here, what you would do is you would plot the expression of those genes against each other, and if you get a distribution like this, it's clear to see that there is a trend line, which you can draw right there. And you can see that some genes are on the trend line, some genes are quite close, some genes are quite far away. But especially considering that this is only two genes, this is a lot of data to be talking about for only two genes if you want to analyze the entire genome. So what you can do is you can apply this function, which is known as the Pearson correlation coefficient function, and that transforms all of this messy data into one nice neat number known as the Pearson correlation coefficient. The way I would encourage you to understand this is it's essentially the likelihood of being able to predict the expression of gene X from gene Y, and of course from gene Y to gene X because they're the same. Now, you can have different types of correlation. You can see that strength of correlation is basically based on how close you are to that trend line. And the more random your distribution gets, the weaker your correlation gets. Of course, you can have negative correlation as well. It's important to mention that negative correlation does not mean negative expression. It just means that when gene X is upregulated, gene Y is downregulated. And another important thing to stress is that correlation is expressed on this kind of color scale, where dark blue is is a heavy positive correlation, dark red is a heavy negative correlation, and white is no correlation. So these graphs would turn into these dots. All right, and a correlation matrix looks something like this, where you have a list of genes on the x-axis plotted against itself on the y-axis. You can kind of chop this matrix in half, which is why it's a triangle, again, because x-y interactions are the same as y-x interactions. And stronger correlations, again, have darker circles that are also larger. Weaker correlations have lighter, smaller circles. So this is only about 30 genes, and we decided to do this analysis for all 20,000 genes, which looks like this. If it loads. Okay, there we go. So this is, oh, whoops. Okay, so this is 20,000 genes by 20,000 genes, which is a matrix of 400 million individual correlations right there. Of course, it's chopped in half, so I guess that's 200, but that's still a lot. So a fun fact, the resolution of this image is about a gigapixel, and displaying it properly at full resolution would require almost 16 televisions, which I don't think we have the budget for, right? Probably not. Okay, so just some main observations to note here. Firstly, there's no obvious patterns in correlation apart from like some stripes for related proteins or genes. Secondly, there's about an equal distribution of positive and negative correlations. Again, there are some stripes, but that's just because these genes are named similarly. And thirdly, and most importantly, these correlations are fairly strong, which makes sense. The human body is a very ordered system, and pathways work together to accomplish tasks. So you would expect correlations to be quite strong. But as you saw a little preview of there, the story changes quite drastically when you look at the in-flight dataset, which, if it loads, or if it works, looks like this. So again, there's a stark difference here. It's easy to see that there's a very significant breakdown in correlations all over the genome, and we think this is very, very important. This would imply that the astronauts' bodies are working against themselves and wasting energy, possibly losing regulation, maybe even activating conflicting pathways at the same time, which has been looked at in previous research, but not extensively. And you would expect these correlations to return back to normal after the astronauts return to Earth, but if you look at the post-flight dataset, that is very clearly not the case. So again, the post-flight data set shows a breakdown in correlations. And again, I'd like to stress that this data set is collected 3 months after the astronauts return to Earth, when most of them have stopped complaining of any remaining physiological issues that they experience. So here's all three of these tables together. Again, you can see a very, very significant breakdown in gene pair correlation all over the genome. And again, I'd like to point out that some areas do have conserved correlation, just a tiny bit, and some areas do also have correlation recovery. But again, just overall, there's a very stark loss in correlation. Now, obviously, 20,000 genes is a lot, and it's too many correlations to analyze effectively, so we introduced a differential expression and correlation cutoff, and that gave us a list of 32 genes that are the most differentially expressed and also have the highest change in correlation. So these are those 32 genes. You might recognize some of them, as a large number of them have recently been implied in spaceflight, and a few of them were also presented about in this conference. Obviously, the selection criteria for these genes did include correlation breakdown, but it's interesting to see the stark contrast between the pre-flight and in-flight data sets for these genes. Now, if you move on, the next step was to use these 32 genes for a pathway analysis, and we came up with this table. Here you can see that a lot of the most involved genes here are cancers or carcinomas, which fits in with recent reports that astronauts suffer more more cancer-related illnesses than normal people. But there's still a wide variety of illnesses here, including psychological illnesses like schizophrenia, tendency towards bipolar disorder, and unipolar depression. Now, we wanted to see if these diseases were only specific to these 32 genes, and so we ran a pathway analysis on the entire data set, which again is 20,000 genes large, and we came up with these pathways. So just to explain the format really quickly, we have the pre-flight, in-flight, and post-flight correlation plots here, and these two plots show the change in correlation from pre-flight to in-flight, which is where the loss happens, and the in-flight to post-flight, which is where you would expect any recovery to happen. And I'm going to skip through these very quickly. So this loss in correlation is mirrored in signal transduction, immune system, gene expression, metabolism of proteins, generic transcription, developmental biology, lipid and lipoprotein metabolism, axon guidance, innate immune system, and very descriptively, disease. So I like this part a lot because not only do I get to talk fast and show a lot of pictures, but it also shows how far-reaching and widespread this correlation loss is across the entire genome. So the next question we asked was, why is it this widespread, and could these 32 genes be a factor? So to understand this, we did a network analysis where we created interaction networks for all of these 32 genes. So this is the protein-protein interaction network. As you can see, there's thousands of interactions here. It's a very dense matrix, and it's showing just how interconnected these genes are. And because of this high degree of interconnectedness, we think it's likely that these genes play a controlling role, or at least have a cascading effect when it comes to the physiological issues that astronauts experience. You can see the same story in the microRNA network. Again, this is microRNA to microRNA interactions. There's a very, very dense network, and again, we think it's the likely cause of these genes being important. Now, they're not labeled here, but the disease gene network shows many diseases with symptoms that are very familiar to astronauts. For example, inflammation, joint pain, neuroocular pressure, the list goes on and on. You can see that some genes like CCL2, DPP4, CRHR1 are important because they connect to more diseases and are implied in more diseases than the others, but regardless, all of these genes definitely play a role in some diseases. And so when you do a treatment analysis, which is the logical next step, you come up with these available drugs for these genes. Some of these genes are missing here, but you can see that there are quite a few drugs that actually do relate to these genes, DPP4 especially. And all of these provide possible treatment opportunities because all of these drugs are currently FDA approved. All right, so three main tenets of this research that I'd like to stress again. Firstly, spaceflight is associated with a significant loss in correlation, which can have sweeping and far-reaching effects when it comes to the rest of the genome. Secondly, this loss does not fix itself, at least within 3 months of returning back to Earth, which is long after physiological effects, most physiological effects subside. And thirdly, that this provides a very exciting possible diagnostic, monitoring, and treatment opportunity for astronaut health. Again, this data was obtained from hair samples, which is relatively non-invasive, so it's very useful. So some issues with the study, of course, only 600 people have ever been to space, around 600, and so you're always going to have a low sample size in space studies. In this case, only 10 people. There's a low diversity in that sample as well. 8 men and 2 women were in studies, which is not representative. This assay also needs to be validated, but this can be solved by using multiple samples from astronauts to increase sample size and also using simulators to get more data. We do need to verify that correlation breakdown also happens in simulations. As for next steps for this project, we've identified 32 genes. We believe there might be more of these highly correlated and highly differentially expressed genes, and we need more samples to confirm this. We have proposed, just as an aside, the 10 sample study was the highest number of samples we found that was open as well. So yes, we definitely need more samples. We have proposed an RT2 PCR assay for astronauts to possibly be able to see the correlation breakdown and experimentally validate these results once more. And I would also like to point out that our lab has published multiple papers about gene pair correlation and its usefulness in other fields, especially cancer diagnostics. And with that, I'd like to mention again that the paper we've written about this is currently under review. It is not published as of today, but it is up as a preprint because we wanted to get the data out as much as possible. And before I open up for questions, I'd like to thank COSPAR again for giving me the opportunity to speak at my first conference. Again, as an undergrad, that's a very high honor. Definitely Prof. Yang, my research supervisor for the guidance throughout this research project, the original authors of the study, and the session moderator, and thank you again in the audience for your attention. Apart from these questions, please feel free to contact me if you have any comments or discussions, and I'd be glad to have a discussion with you. Thanks.
Questions?
Very nice talk.
Thank you.
I wanted to make sure I understood the correlation. What was the vector? What was the things you were changing in each of the correlation? Was it the different people for your vector for the correlation?
Are you referring to the three correlation plots that I showed?
Any of the correlation that you show, yeah, those one. Yeah. So what is the when you do correlation, right? Like you said, you have a bunch of points for X and a bunch of points for Y. So
yes.
What are those points? What is your what are you changing between the points? Is that the different
The
people?
points are individual gene expression values. So each of these graphs has, I guess, millions of little dots, and those dots represent, I guess, the correlation values for whatever gene is in the X and Y axis.
Right. So it's the correlation between gene X and gene Y, for instance.
Yes.
But to get a correlation, you need multiple values. So
yes.
Where do those multiple values coming from? Is it the different? You were looking at a study, right? So you had
yes.
like
those multiple values are coming from
the different people.
Yes, the different people, and all of their values are, I guess, put on the same plot, and then we take the line of best fit and apply the correlation coefficient function.
Okay,
Yep.
because there's different way of doing it. You could also use as a vector your pre-flight, in-flight, post-flight. So then you have
Yes.
3 values, or you could even have more if you want to add all the people and all the changes. But I think it's a very elegant idea. It's a different way of doing things. So I thought we should talk more about this afterwards.
Definitely. Yeah.
Any other questions? So, do you know why? I mean, I'm very naive about these matters, but the original study, what was the advantage of like studying hair follicles as opposed to, let's say, other cell types? Is it ease of collection, or?
I think the objective of the original study was mainly focused on hair. They wanted to look at the diagnostic potential and gene expression change in hair, or of specific genes in hair,
Okay.
when it comes to tissue-specific gene expression.
I see.
Yeah.
I see. I see. All right. All right. So, I'd like to end this session by thanking the speakers and the attendees, and I wish you a great evening.